Alright, so here we are. This is our lecture on enzymes. These are the objectives. I know there's quite a few of them, but they're really specific. The main thing you need to know is what an enzyme is, what its main function is in living organisms, and how it is so essential. And then also some very special characteristics of enzymes. Alright, so let's talk about chemical reactions. This should look very familiar when we look at this graph. We just talked about it in our last video. As we go through a reaction and we go from reactants to products, there's a certain amount of energy that is required. Okay? So we need to input energy into the system, and remember that energy being inputted is to break bonds, and then energy is given off as the products are made, and if there is more energy given off than we had at the beginning, then we're going to call this an exothermic reaction. Okay? Uh, but regardless whether it's exothermic or whether it's endothermic and we end up here with the products, remember they all require this amount of activation energy. So, what can a catalyst do? Here's a new term. Catalysts allow reactions to happen faster, and they do this by decreasing the amount of activation energy that is required. So let's zoom in here on this diagram. Okay, so let's take a big wink wink, nudge nudge, double star this diagram, because you will need to know this from now through any college biology, any AP, any science you need to know, going into physics, everything like that. What we're showing here are two different lines to represent two different ways a reaction can happen. So let's take a look at the gray line here. Notice we have our reactant energy starting at this level. We need to go up the hill, and when we get to a certain point, when we've put enough activation energy in, then everything falls down and our product energy is much lower. Okay, so this arrow represents the amount of activation energy that is required for the reaction to happen all on its own. Now looking at the other line, this is what happens when we add an enzyme to the system. Notice we are, there we go. Notice that we're still starting with the same reactant energy, but this time the hill is a lot easier. Okay, not quite as steep, so we're going to be able to get up to our need for activation energy much faster, which means we're going to be able to get down to the product energy much faster. Important thing to note here as well, if you look, the product energy has not been changed either. So the reactants and the products are unchanged as far as the amount of energy. What is different is that we lower the amount of activation energy that's required to get it to start. Okay? So the idea is it's sort of like pushing a snowball up the hill. The steeper the hill, the more energy you have to put into it. Wouldn't it be nice if someone came along and cut down that hill so it wasn't so steep? You would be able to push the snowball much faster and get it over the hill. This PowerPoint has a number of links that are on the bottom of the slide. Don't worry, we have put all of these links up on the website under the A2 unit links, and each of them are numbered. So this one, for example, this is called enzyme OV link number 1. Okay? So that's what a catalyst does, speeds up chemical reactions. And in living things, there are proteins that act as catalysts, and we call these things enzymes. And so that's what this entire PowerPoint is about. So an enzyme is an example of a catalyst, and all enzymes are proteins. So let's just take a look at a little diagram here. This is a very simplified version because proteins that are enzymes, they can be very large and complex. So sometimes we like to sort of cartoonize the idea so that it's easier to understand. So this green piece on the side here, we're going to say that that's our enzyme. And enzymes have active sites, okay? Whenever we have products that have, whenever we have reactants that are being turned into products, the enzyme and the reactants physically attach to one another, and the active site is where that happens. So with the reactants, that are interacting with an enzyme, we're going to call them a substrate. Okay, so here is a reactant. All right, let's say it's a disaccharide. It's going to attach to this enzyme, and when they attach together, they form what's called an enzyme substrate complex. Then the enzyme is able to do its job, and in speeding up the chemical reaction, we have products. Okay, so for example, like I said, if this was a disaccharide, right, it had two simple sugar units that linked it together to be a polymer. The enzyme assisted in the chemical reaction by allowing it to happen faster, and it broke that down into two separate simple sugars. So here's a good question. Would this reaction be an example of dehydration synthesis or hydrolysis? It would be hydrolysis because we are taking a large polymer and breaking it down into its subunits or into monomers. And then please note that at the end of this, the enzyme leaves unchanged and reused. So we're going to touch on that in just a second. Key characteristic number 1 of enzymes. Enzymes are specific. Okay, so they fit. We used to say they have like a lock and key fit, and what that means is there is one enzyme out there for each kind of substrate, and I mean there are many copies of that enzyme, but there's one enzyme that has that specific shape in order to fit that one substrate. So very much like there are many millions and billions of different keys, and there are locks that go with them. Only one key is going to open one lock, right? Okay, so my front door key is not going to unlock your door. It's specific to just my door. Same thing. So enzymes are going to be specific and can only fit to the substrate that they are meant to fit for. Okay? Another thing, enzymes are unchanged and reusable from a reaction. So you saw this before, but here's another diagram to show it. This big gray guy here, this is our enzyme, all right, and we're going to say that this is our substrate. It attaches at that active site, okay, and then the enzyme sort of hugs in because it says, oh, I recognize you. You are the substrate that I work on. And it sort of changes a little bit of shape, just slightly, in order to better fit to those substrates. And then when it forms that enzyme substrate complex, that's going to allow the products to form. Note what hasn't been changed is the enzyme, right? The enzyme has stayed the same. So it gets reused. So it sort of moves all around in this, the cytoplasm or wherever it is. It finds its reactants or it finds its substrate, binds to the substrate, produces the products, then goes out and finds more of its substrate, produces the products, and finds more. So they're reusable. They are not changed in any way by a chemical reaction. Enzymes can be affected, however, if we start playing with environmental factors. Okay? Enzymes are very sensitive to changes in pH and temperature. So there can be a situation, for example, where enzymes can be too acidic or they could be too hot. And this is not good because what happens is that big, large enzyme has a specific shape, right? Remember that active site? There was a specific shape that was needed so that the substrate could bind to it. If that shape comes undone, okay, as temperature rises or as pH changes too much, what's going to happen is that specific shape is going to break down, and we call that denaturing. So when you look at a protein, basically what you have is it's just a big long mess of all these intertwining chains of amino acids, and they're all being held together by bonds. Trying to think of a good example of what it would be like. It would be sort of like if you took a lot of strings of beads, okay, and you had this string of beads and you had like thousands of them, and you attached them to each other by putting a little dab of glue on a bead on this string, and then a little dab of glue on a bead of this string, and attaching them together. and so they make this really cool shape. When you would apply too much heat, then the glue would melt and your shape would come undone. That's exactly what's going on with enzymes, okay? They're going to start to denature or lose their shape as pH or temperature changes. And if they lose their shape, then that's going to seriously affect their function. Exactly, right here. Okay, so here's a really good diagram to show you an example. All right, here's an enzyme, and these happen to be the two substrates that it works on. So the enzyme goes to bind to the substrate. Notice how that active site is so lock and key to the substrates, okay? Now we add heat. What happens to the enzyme? It changes shape, okay? And now when it changes shape, what has happened to the active site? It has been altered in such a way that these substrates cannot bind anymore. And so the substrates aren't able to bind, and that's going to be bad because that means that the enzyme cannot do its job. Enzymes are so essential to living things that if chemical reactions happened without enzymes, they would happen so slowly that we wouldn't be able to sustain our cell functions. So even though an enzyme is sort of like a turbo booster pack on a chemical reaction happening, if we didn't have it, it wouldn't work. We wouldn't be able to survive without them. So we need that turbo boost. So this brings back one of the themes, looking at homeostasis, needing to be able to maintain an internal environment. Obviously, for all living things that have enzymes, we need to keep a constant pH and a constant temperature in order to maintain our enzyme function. So two themes there. We talked about homeostasis, and we talked about the fact that when you change the structure of something, you alter its function. So again, structure and function are related. Just a little hint, as you're reading things, you want to zero in on enzymes. Most enzymes end in an ASE ending. And what that means is it is an enzyme that does blank. So for example, protease, okay, there's the ASE ending. That's an enzyme, and it tells you what it breaks down. What does that remind you of? Protein, right? So you have lots of different types of proteases in your body that are very specific to one type of substrate. They can be reused, unchanged, and if you start playing around with pH or temperature too much, they can be denatured. Okay? And then, these are just some other examples here. The DNA polymerase, it polymerizes or it joins together monomers that make DNA. And this is called ATP synthase. And basically what it does is it helps to synthesize or produce ATP molecules. So enzymes, very important, very essential, very picky. All right? Make sure you know those characteristics, and if you have any questions, please ask your teacher in class.